Hello students, welcome to the class. Today's our topic is the origin of first cell and introduction of prokaryotic cell. How did the first cell formed? We will discuss this in depth in evolution playlist. In short, proteins and RNA were thought to be the first macromolecules produced. Only RNA, capable of directing its self-replication, acting as both template and catalyst, means it can replicate by itself using self as template and enzyme for its replication. RNA world existed for a period of time. The first cell is thought to have arisen when RNA became enclosed in a phospholipid membrane and was able to self-replicate. There are two types of cells, prokaryotic and eukaryotic cells. In this class, we will mainly talk about prokaryotic cell. Prokaryotic cells. These cells are simple type of cells. They don't have nucleus membrane, means uncovered nucleus. Have no membrane-bound organelles. Single circular DNA genetic material, which is quite different from eukaryotic chromosomes. Nucleoid region, center, contains the DNA, surrounded by cell membrane cell wall, which is made up of peptidoglycan. Contain ribosomes which doesn't have membrane. In their cytoplasm to make proteins. Includes bacteria. Present-day prokaryotic cell belongs to two groups, archaea and the eubacteria, true bacteria. Archaea live in many harsh environments. For example, methanogens. These archaea produce methane gas as a byproduct of their metabolism. They are found in anaerobic environments like marshes, hot springs, and the guts of animals, including humans, halophiles. These archaea thrive in extremely salty environments. Thermoacidophiles. These archaea tolerate and even prefer high temperatures and acidic conditions. They can be found in hot springs and other extreme environments. Eubacteria populate many places. Bacterial shapes vary, as does their size and DNA content. For example, cyanobacteria are sometimes considered algae, where the term algae is now reserved for eukaryotic organisms. They also derive their energy through photosynthesis, but they lack nucleus or membrane-bound organelles, such as chloroplast, components of prokaryotic cells. All cells share four common components: plasma membrane, an outer covering that separates the cell's interior from its surrounding environment; cytoplasm, which is a jelly-like cytosol within the cell in which other cellular components are found; DNA, the genetic material of the cell; ribosomes, which helps in protein synthesis. Some other components are pili, flagella, fimabrae, capsule, cell wall, mesosome, chromatophore. So the question arises, what are the fundamental components of prokaryotic cells? First structure is nucleoid. Nucleoid means nucleus-like is an irregularly shaped region within the prokaryotic cell that contains all or most of the genetic material. It is not surrounded by nuclear membrane. Prokaryotic DNA and DNA-associated proteins are concentrated within the nucleoid region of the cell. In general, prokaryotic DNA interacts with nucleoid proteins, NAPs, that assist in the organization and packaging of genetic material. Most prokaryotes like E. coli contain a single circular DNA molecule that make up their entire genome. Plasmids is another important component of prokaryotes. Prokaryotic cells may also contain extrachromosomal DNA, which is not part of the genomic content. This extrachromosomal DNA is found in plasmids, which are small circular double-stranded DNA molecules. Plasmids are more commonly found in bacteria, however plasmids have been found in archaea and eukaryotic organisms. You will understand the importance of plasmids in genetic engineering in the upcoming biotechnology classes. Ribosomes. Prokaryotic ribosomes are also called 70S ribosomes because they have size of 70S, whereas eukaryotic cytoplasmic ribosomes have size of 80S. The S stand for Svedberg unit, a measure of sedimentation in an ultracentrifuge, which is based on size, shape, and surface qualities. Ribosomes in each domain, bacteria and archaea, is structurally different. Ribosomes help in the protein's synthesis in cellular life through to the process of translation. Prokaryotic ribosomes 70S are composed of two subunits, the 30S, the small subunit, and the 50S, large subunit. and each of which are composed of protein and rRNA components. Another structure is endospore. Bacterial cells are generally observed as vegetative cells, but some genera of bacteria have the ability to form endospores. These are structures that essentially protect the bacterial genome in dormant state when environmental conditions are unfavorable. Vegetative cells and endospores have big differences in their properties. Let's compare them. Vegetative cells are sensitive to extreme temperatures and radiation. Endospores are resistant to extreme temperatures and radiation. Vegetative cells absorb gram stain and are gram-positive. Endospores do not absorb gram stain, only special endospore stains. Vegetative cells have normal water content and enzymatic activity. Endospores are dehydrated and have no metabolic activity. Vegetative cells are capable of active growth and metabolism. Endospores are dormant in nature and show no growth or metabolic activity. The process by which vegetative cells transform into endospores is called sporulation. It generally begins when the nutrients become depleted or environmental conditions become otherwise unfavorable. There are 6 steps in the spore formation. First, the DNA replication begins. After that, membrane forms around the DNA. Spore forms the additional membranes around and formation protective cortex around the spore, as well as formation of protein coat around the cortex. After all this process, the spores are released. Sporulation begins following asymmetric cell division. The spore becomes surrounded by double layer of membrane cortex and protein spore coat before being released as mature endospores upon disintegration of the mother cell. Summary: in today's class, we have learned about the cell origin prokaryotic cell and its properties, prokaryotic cell fundamental components, in which we have covered nucleoid plasmids, ribosomes, and endospores. In the next lecture, we will cover all the remaining prokaryotic cell structure, such as plasma membrane and its modification. Capsules and slime layers. After that, we will cover flagella in detail and pili. In last, we will understand the experiment which helps in bacterial cells differentiation, which is quite popular and named on the scientist who discovered the method. If you know the experiment, comment down below. If this video has helped you in clearing your concepts, then please like and subscribe. Thanks for watching.